Our next speaker, Alec Paciorkowski, is a senior instructor in neurology, biomedical genetics, and pediatrics here at the University of Rochester. His lab is focused on understanding the genetic causes of developmental disorders of the brain, including autism, childhood epilepsy, brain malformations, and movement disorders. The project in his lab used a number of tools, including molecular genetics, developing in vitro models, and the analysis of next-generation sequencing data.
Well, thank you very much for the introduction and the opportunity to share some of our more recent data with you, as well I'm hoping some of the rationale behind our approach. One of the issues that is increasingly becoming obvious is we can generate large amounts of sequence from our research subjects, but the interpretation of that continues to be somewhat where the cutting edge continues to lie in the field. In terms of autism, our group kind of backed into autism research from the side door, as it were. My group studies infants who present with severe early onset epilepsies. As you can see in the slide here on the left are representative EEG tracings from specific epilepsy syndromes, which have trademark features in terms of the seizure morphology. But one thing that we're learning from the underlying biology more and more here on the bottom is that there is shared biology underneath most of these epilepsy syndromes, such that if you track the natural history of children over time, a baby at birth may present with Ohtahara syndrome. By 2 or 3 months of age may progress to infantile spasms, and then later on in childhood may have Lennox-Gastaut. And all of these are mediated by families of genes which interact with each other. One of the other things that's been present in the literature really since the 1970s has been the observation that many of these children do go on to develop later onset autism, that they are epilepsy patients when they're infants, but then when they're restudied at older ages, they do have many autistic features, and that's something that's been reported on increasingly in our literature. So there is this very clear link between early infantile epilepsies and the later prevalence of autism in this population. Now, my lab is interested in discovering the new genetic causes for these disorders, and to do that, we use whole exome sequencing, as you've heard several times today. This is a methodology that is becoming easier, but still, when you take a step back and look at the handling of data, there definitely is room for challenges still, especially when you consider that the average individual has about 3 million single nucleotide polymorphisms. Those are areas that diverge from the reference genome. Now, in whole exome sequencing, we're only looking at the coding regions, or the exome, so that really gets us down to about 20,000 here in this schematic, 20,000 variants, which is still an acceptably impressive number. But then we really want to focus on those changes in the coding regions that cause amino acid substitutions, and that brings us down by another 10,000. Then we're looking then at the pedigree and whether these are rare variants in the general population, and ideally we will get down to the hundreds, and then eventually through various filtering, especially by pedigree and other population studies, getting down to one or a couple of candidate genes. So we start with many, many, many thousands, if not millions, working all the way down to one variant is ideally how this should go. And when we do sequencing of this nature in our populations, that is what we find. We do find solutions for many families where they previously did not have a diagnosis. This past month, we did whole exome sequencing in a group of children with something called malignant migrating partial seizures of infancy, which is about as bad as it sounds. And we found 2 children with mutations in a gene called KCNT1, which was described in a first publication a year ago by a group from France, and this is now a confirmatory population from our lab. Additionally, we just published a new gene for infantile spasms called KPNA7. This is an autosomal recessive gene, and we'll talk a little bit more about that one later. And the most recent finding over the past couple of weeks has been a mutation in this gene called SIK1, which is found in a child with Ohtahara syndrome. We've just identified a second patient with a deletion of that gene. This is also interesting because it enables me to introduce a theme that we'll go back to throughout this talk, where this gene is involved in a biological pathway with other genes that are already known players, already known to be causative, both in infantile spasms, infantile epilepsies, as well as developmental disorders like autism. So this is going to be a common theme we'll come back to that helps us reach this solution. Because that challenge of the analysis is, even if you can get your numbers of variants down to 100 or 50, if you're dealing especially with a de novo pedigree, that's still quite a large number of variants in genes that are plausible candidates. This is a typical pedigree that we're dealing with in autism as well as in most infant onset epilepsies. Usually you find there is one affected child, and there may not be another affected individual in the entire family. We then have a bunch of criteria that we are interested in. We are hoping that the gene member that we're looking at is going to be a member of a known pathway, a known biological pathway. And that's going to be the real criteria I'll spend most of our time talking about, is how to curate and organize the information that's known about genes in the human genome to identify which genes are actually members of known pathways, because that has been to date a very rich source of new discoveries. Now, looking at the biology, we really find that three main pathways are hit over and over and over again in both autism and infantile epilepsies. These are a sampling of genes over here for the infant epilepsies, many of which have autism as part of their later clinical features. And these are all genes that are involved in synapse development and function, whether they're ion channel genes such as potassium or sodium or calcium channel, or whether they're proteins that reinforce the pre- and post-synaptic membranes. This is certainly a rich source of new genes. And there's about probably 2,000 to 2,500 different proteins that go into making a neurologic synapse. The next group is one that's emerged as really exciting and surprising. And these are genes where there are post-zygotic mutations or mosaic mutations in the brain in genes in the mTOR RAS growth pathway. These are omnipresent, ubiquitous regulatory pathways that are also very important in cancer. And what's exciting about these is that there are already targetable pathways that are being researched, and we're hoping that there will be new epilepsy treatments coming in the near future out of drugs that are already in development for this pathway. And the third one are a group that I've been working on, are forebrain transcriptional regulators, among them KPNA7, which I talked about. This is a protein that carries transcription factors into the nucleus. And if you have mutations in this gene, you abrogate the ability of the protein to carry transcriptional regulators into where the DNA lives in the nucleus. So if you remember all of these three, we're going to come back to these again and again. This is more looking on the genotype side of things for our data. But then we have to ask, well, what about the phenotype side? Is there any way that we can link the two? This is where my group spends quite a bit of time as well. If we focus on the center of the slide up here where it says classical autism, we see that many of the canonical behavioral traits that we would associate with children with autism, restricted interest, repetitive behaviors, language disorder, and impaired socialization, there's a certain clinical impairment associated with this triad. Whereas we all know people who may have one or two elements of these but are able nevertheless to function. So there certainly seems to be a continuum of these traits throughout the general population. Now, my group has been mainly working, as I said, from a side door, as it were, to autism, where these are conditions I've always called autism plus, really for lack of a better term, where the children certainly do have elements of this triad of impairment, but then when you look at other traits such as named epilepsy syndromes, dysfunction of the autonomic nervous system, a movement disorder, a sleep disorder, the list really goes on and on. There are just a staggering number of neurofunctional domains that are impaired in these children. Now, if you lay these out on a line and begin to say, well, how do they interact with the genotype? That's really the question that we're looking to investigate. We have the synapse development and function genes, again, a few sampling for autism, cellular growth as well, and the same pathways, forebrain transcriptional development. The question is, are there particular patterns of traits that will map to these different pathways, and can we use them to make appropriate diagnosis by recognizing clinical patterns and identifying pathways that are impaired in those children? Can we use them for prognosis for the families? And increasingly, can we identify treatments, which is, I think, where all of this research inevitably is heading, hopefully not in the near future, not in the distant future, but in the near future, where we can recognize different patterns from our phenotyping and infer from them whether or not certain genetic patterns may be present or not. So what have we done to bring this about? We looked at 10 disorders. These are disorders I again call autism plus disorders. Some of them are more well known than others. All of these involve severe cognitive impairment and developmental delay. And we extracted from the literature programmatically, basically all of the traits, phenotypes, descriptors, clinical findings that are out there, and assembled essentially a global developmental inventory of all of the identifiable, nameable neurofunctional domains that are impaired in these childhood disorders. And this really became sort of an ontology of neurobehavior. And it's a long list. There's over 100 elements that we do ascertain. We do this there's some structure. Things are organized by ambulation, autonomic nervous system, awareness of self and environment, on and on. We don't need to go through all of them, but when we collect that data and apply them to genotype known groups, we can begin to recognize patterns for certain disorders. And I'm going to share with you one data set from a group that we've worked with extensively. These are children who have mutations in a gene called FOXG1, which is a forebrain transcriptional repressor that's important in early dorsal ventral differentiation of the brain. And these children are phenotypically very interesting because their phenotype depends on their genotype in a very, very fascinating way. If you focus on panel C, this is chromosome 14 laid out on its side. FOXG1 is circled right there. All of the blue bars are genomic regions where there are duplications, where these children have an extra copy of FOXG1. The red bars underneath are deletions. So those are children who all have a missing copy of FOXG1. And then below here in G, we have a number of children who have intragenic truncating mutations, which serves as a loss of function, essentially deletion as well. Now, the children up at the top who have duplication or an extra copy present with an early infantile epilepsy. They respond to treatment. They never have A, yes, right up here at the top, sorry. They respond to treatment, and they don't ever have recurrent seizures. We followed them now at age 9, 10, and they have an autistic phenotype, and when they're seen, they're almost all diagnosed with classical autism. Children with deletion down at the bottom have a much more severe neurobehavioral phenotype. They have a choreiform movement disorder. They also have a morphologic abnormality of something called the corpus callosum, which is this structure right here in the middle of the brain. And this is normal, and this is children with FOXG1, where the head size is also small, and they also have hypoplasia of the corpus callosum, which we'll talk about in a transition in just a minute. So they're very different depending on their genotype. And what we've done is we've applied this structured quantitative way of collecting behavioral data to both of these populations. And if you run it through R, you can generate some nice images where you see at the top, FOXG1 duplications are much rarer, FOXG1 deletions are here. But you can see that there are significant differences. First of all, children with deletions of FOXG1 simply do not walk, whereas the ones with duplications tend to get around just fine. You also see that there are areas where there are core similarities in functional hand use, fine motor movements, language, and certainly social reciprocity that are all impaired, whether you have a deletion or a duplication. And so this is really a quantitative way that's quite novel to approaching these disorders and understanding the differences between them. Now, if you add in girls with Rett Syndrome, which we're currently doing, this is just some of our preliminary data, so we don't have quite the numbers yet, but we can start to see that there are some themes. We know girls with Rett tend to walk around quite well, but there are also important differences in terms of breathing, where there is a known breathing abnormality in girls with Rett Syndrome. And they do have the same core impairments that I've highlighted here. Darker colors indicate more impaired in this type of image. So moving from there to another way of looking at phenotype, I wanted to just quickly talk about agenesis of the corpus callosum, which is this midbrain structure that I talked about. This is normal. These are children with FOXG1. If you look at children in the general population with agenesis of the corpus callosum, we see that there are an increased number of autistic traits in those children. One thing that our group recently published is a large 250 samples of chromosomal copy number microarrays of children with agenesis of the corpus callosum, and discovered that a significant number, some 10 to 12%, do have de novo deletions or duplications in the genome that are pathogenic. So there are genetic markers for agenesis of the corpus callosum. What you need to know about areas of the genome where there are deletions and duplications is that there are significant genes inside these genomic regions, such as FOXG1, for example, that presumably drive the pathogenesis. And when we're curating our gene content, we need to be able to identify these genes to know whether or not they're actually the disease-causing gene. Looking at genes that cause agenesis of the corpus callosum, this is both human and mouse data. The larger type indicates a greater significance of association. We see that there are several pathways, biologic pathways, for example, axonogenesis. This is forebrain development, mitochondrial function. There are 7 of them that are highly enriched among our genes that are known to be associated in both humans and mice. And we begin to use that to curate the data from our copy number, information from our human subjects. And this is essentially genomic data arranged in a circle. Each bar here is a human chromosome in different colors. And over here are the different functional pathways I talked about, axonogenesis, ciliary function, etc. And we see that the blue areas are the areas in the genome where there are de novo duplications, red are de novo deletions, and within them are genes that have members that are enriched for some of these functional pathways. So these are ways we can begin to identify data that helps us know which genes may be pathogenic or not. So the question is how to organize this information in a way that we can actually use it in the lab when we're doing gene discovery. Our group has just completed this publicly available tool called the Developmental Brain Disorders Database. It's hosted online here at Rochester. And essentially, this is an online repository of gene phenotype associations. So just like what I showed you, when we look at genes for agenesis of the corpus callosum, we want to know whether there's a real association from the literature and how strong that association is. We've looked so far through our group of curators at over 800 gene phenotype associations, and we've applied a level of evidence score, which is way over here in the right column, 1 being a higher level of evidence and 3 being lower. Each page, for example, will have information about the particular developmental brain phenotype and also which genes are associated with that phenotype and the levels of evidence. When you do this kind of work, you also can do some other types of data analysis and look at really what the landscape of what I call the neurogenetics knowledge base is. If you stretch out all the developmental brain phenotypes here on this axis and the number of associated genes here, well, not too surprising, you see that there are a number of genes where there are, a number of phenotypes where there are many, many genes associated. And then for the junior faculty who may want to make discoveries, you may want to live over here where there really isn't as much knowledge. In fact, a few phenotypes where there are no known genes associated at all. But most phenotypes have around 10 associated genes. You can begin to look at linkages. This is kind of one of the last slides that I'll show. Linkages of what genes that are associated with a given phenotype may have in common. So if you're a gene for microcephaly, which is a rather generic developmental brain disorder, you have a connection here in green with microcephaly. These are all in blue, all of the genes that are known to be associated. But you'll also have a secondary association with other phenotypes shown here in pink. And you can see that if you're a gene for microcephaly, you may have many other associations with developmental brain disorders. If you go more specific to something like molar tooth malformation, which is a birth defect of the brainstem and cerebellum, you have fewer connections to other developmental brain disorders. You're more likely to be specifically associated with that birth defect. And then looking even more specifically at something like retinal dystrophy, you see that if you're a gene for retinal dystrophy, it's unlikely that you're going to be causing other developmental brain malformations. So what does autism look like? Not surprisingly, it's very well connected to other things like epilepsy, intellectual disability, as well as several established brain malformations as well. Okay. So the way we've implemented this is my last slide in the lab as sort of a tool that we're due to release sometime later this month, is putting all of this together in a way to actually handle whole exome data in a more efficient way, because most of the labs that are working with this type of data do get to a point where they can no longer programmatically handle their curation, but have to manually begin looking things up. If you've got 50 genes for a family, that's a lot to look up. So we have come up with something we call Solve Brain, which is a software that applies a variety of pedigree and quality control filters, passes all of our whole exome data through the Developmental Brain Disorders database, because if we're dealing with a family that has a mutation in a known gene, I want to know that really quickly. And this certainly helps us by flagging those families. So then you get, if not, you get a prioritized variant gene list, which then the user can then go to the web interface, paste in the gene list, and then it automatically gives you links to all of the annotations that we have routinely used, which are a variety of tools that help us know as much as possible from very disparate sources. And this is part of the issue with this curated knowledge base, is that the information is in several different data sets and really requires an organized way to get at it quickly. And this tool helps to provide that. So in summary, I hope I've shared with you some evidence that there's an underlying biology for autism and related developmental brain disorders, and the recognition of new discoveries depends a lot on leveraging and curating knowledge that's actually already out there, but it's in very disparate sources. And right now, we're still using recognition of shared genetic pathways as a powerful strategy to make new discoveries. And I have multiple individuals to thank, not only in my lab, child neurology here, Jonathan Mink, Heather Adams, our autism research community here at U of R, which is vibrant, Susan Hyman, Tris Smith, Louisa Benetto, and Chris Sajal, as well as our research families, funding sources, and collaborators at other institutions. Thank you for your attention.
Hi, what the next speaker come set up? Okay, well, we're having some questions. Go ahead. So I have a quick question.
Sure.
When you show the picture of autism, so it's a very complicated condition, do we have any sense of how close we are to knowing all of the genes that are involved?
I think we're far from really understanding. There are some families where there appear to be rare variants that are severe, and there are other families where there may in fact be multiple genes where there are changes, and that's really the next horizon, is identifying those families where there may be multiple not only genomic effects but environmental effects as well. So I think it's just like peeling an onion away one layer at a time, and it depends on your annotation of your data how successfully you're able to peel away each layer. There are about 36 genes that have high levels of evidence to cause autism. That's all. So there's surely more.
I have a much less important question. When you mentioned SIK1, you had something of a Venn diagram. Pointing into that Venn diagram was very tiny print the word cocaine.
Yeah, so one of the early studies that identified the transcriptional pathway associated with that gene was using a cocaine-induced gene expression model in rats. So that's the best image I could pull at the time. Good eyesight.
I think we have one more question.
Sure.
When you think about something like autism that may have 36 genes that can play a role or that are associated with it, how do you get to cause and effect when you're thinking about a complex multi-genetic?
So those 36 genes are genes like the Fragile X gene, where there are clear in vitro neuronal studies, and there are also reproducible animal models where the animals with the mutation in that gene have autistic features, and where for more than a decade there have been human studies showing that reproducibly recurrent mutations in that gene cause autism. So those are where their causation is clear. I think there are less than 5 genes where that causation is as clear as that. The rest of the ones on that list are in varied states of disarray, of lack of knowledge underneath that. So there's a lot of work to do.
Okay, thank you.
Sure.